civilians involved. So Merck, Sharp, and Dome was one of the original companies. And this is just an article called The Birth of the U.S. Biological Warfare Program. And it just talks about the government's files and who was involved from the beginning. I found much of this very interesting because I worked at Walter Reed Army Medical Center in Washington, D.C. for about 2-3 years. So I was there as a graduate student, but I'm working in this environment knowing full well that their agenda is to create things for destruction. I mean, that's what the Army is for. Walter Reed Army Medical Center is also the place where the presidents are treated, where certain governmental personnel are receiving their treatments. So it's a pretty secure place. But I noticed that many of the places that have been associated with the creation of these substances of chemical biological warfare came from Fort Detrick, Maryland. Fort Detrick, Maryland is the hub for Walter Reed Army Medical Center. That's where I was getting my paychecks from, Fort Detrick, Maryland. So I started to question, now why am I working up in here if I know full well that I shouldn't be involved in issues, research that may have an impact on us as a people? So I began to do some reading while I was there, and that's when I had an opportunity to really kind of uncover the government's role in some of these serious issues. The search for the ultimate weapon. The military has several times expressed its fantasies for new biological weapons. Some of these images are chillingly suggestive of a microbe that would cause AIDS. So prior to the word and term acquired immune deficiency even coming aboard, there were thoughts about how do you create something that could do that. In 1969, a military official testified before Congress. Within the next 5 to 10 years, it would probably be possible to make a new infective microorganism which could differ in certain important respects from any known disease-causing organism. Most important of these is that it might be refractory, that is resistant to the immunological and therapeutic processes upon which we depend to maintain our relative freedom from infectious diseases. In a book on CBW written just before the identification of AIDS, 2 authors commented on this testimony. The possibility that such a supergerm may have been successfully produced in a laboratory somewhere in the world in the years since that assessment was made is one which should not be too readily cast aside. This is not an entirely academic speculation. In 1968, Porton Down, the British Army's Biological Warfare Laboratory, and Fort Detrick collaborated in the successful transfer of genes between different strains of plague. The research was done for purely, they say, defensive purposes. It goes on to say that added to all this suggestive evidence is the particular history of the Army's Fort Detrick facility in Frederick, Maryland. It is here some have charged that the Pentagon may have developed AIDS purposefully or accidentally. This center was called the Army Biological Warfare Laboratory until Nixon's 1969 order banning CBW for production. Fort Detrick was then the leading center for development and testing of biological weapons. So we now have the physical evidence that these things were created here, and now you have the governments all worried and concerned about other countries, other militaries, other people getting a hold of something they created. Well, it's inevitable that it could occur, but the point is, there's only one diabolical mind that's ever used it and done it. If you even talk about the nuclear bomb, there's only one diabolical mind that's ever dropped a nuclear bomb. And we're always portraying and talking about other people being the problem, and here we have a diabolical mind here that we're not really paying attention to. These 2 things, as we try to move on, about the higher form of killing. It's a book written in 1982 by Robert Harris and Jeremy Paxman. It says A Higher Form of Killing, The Secret Story of Chemical and Biological Warfare. Just a few pieces from it. The San Francisco test was only one of many, talking about documented evidence on and on about how they actually use things and manipulate things in the environment and people weren't really paying attention to. In 1951, American Navy personnel deliberately contaminated 10 wooden boxes with Serratia marcescens and one of the things we just talked about, Aspergillus fumigatus, before they were shipped from a supply depot in Pennsylvania to the Navy base in Norfolk. So this is corroborating the story now from another source of information. The tests were designed to establish how easily disease might be spread among the people employed to handle the boxes at the supply depot. Of the 3 infectious bacteria, this Aspergillus fumigatus had been specifically chosen because black workers at the base could be particularly susceptible to it. Another piece which implicates the Fort Detrick place and kind of talks about Georgia here. It says Fort Detrick scientists discovered a Trinidadian who had been infected with yellow fever in 1954 and had later recovered. So you know Trinidad is African people, right? They took serum from the Trinidadian and injected it into monkeys. From the monkeys, they removed infected plasma, into which they dropped mosquito larvae. The infected mosquitoes were then encouraged to bite laboratory mice and pass on the disease. This ingenious technique of public health research in reverse worked. The mice duly contracted yellow fever. Laboratories were built at Fort Detrick, where colonies of the Egypti mosquitoes were fed on a diet of syrup and blood. They laid their eggs on moist paper towels. The eggs would later turn into larvae and eventually into a new generation of mosquitoes. The Fort Detrick laboratories could produce 500,000 mosquitoes a month, and by the late 50s, a plan had been drawn up for a plant to produce 130 million mosquitoes a month. Once the mosquitoes had been infected with yellow fever, the Chemical Corps planned to fire them at an enemy from cluster bombs dropped from aircraft and from the warhead of the Sergeant missile. I remember first moving down here in 92. In 92, 93, mosquitoes were just horrible around here. I mean, they were the tiger mosquitoes. They actually had little black and white stripes on them. I don't see the major thrust or contamination of mosquitoes today. It could be because of a variety of factors, but I'm just looking at that period of time. It wasn't even enjoyable to go out and play, sit out on the deck, go to a cookout, because mosquitoes were everywhere. So you can see how that would be a perfect plan to get people, because you have this contagion in these little organisms that you're not even paying attention. You're just swatting it. You're just swatting it, but little stick and prick, you don't know what then happens in terms of the future with your body. To test the feasibility of this extraordinary weapon, the Army needed to know whether the mosquitoes could be relied upon to bite people. During 1956 that is, they carried out a series of tests in which uninfected female mosquitoes were released first into a residential area of Savannah, Georgia, and then dropped from an aircraft over a Florida bombing range. Within a day, according to a secret Chemical Corps report, the mosquitoes had spread a disease of between 1 and 2 miles and bitten many people. The effects of released infected mosquitoes can only be guessed at. Yellow fever, as the Chemical Corps noted, is a highly dangerous disease, at the very least, causing high temperatures, headache, vomiting, blah, blah, blah. So this thing about the yellow fever, if you go to any book on microbiology and pathology, happen to read this about this yellow fever and the actual organism that creates it. It says, it is one of the most infectious of the fungus diseases. Now this is an old school book now. I think we might have still been Negroes here. This is 1972, so we're still probably Negroes and blacks here. It says, in the primary form, the lesions are confined to lungs, giving pulmonary symptoms of varying severity. Sometimes there is cavitation in the lungs. As a rule, the infection ends in recovery, but in a small percentage of cases, the process spreads from the lungs to produce the progressive form. This occurs more frequently in Negroes and the more darkly pigmented races than in Caucasians. So you don't just take that lightly. Just because they're saying that does not mean that they're not using that to study, to use as a weapon. In the progressive form, the disease spreads to the skin, subcutaneous tissues, bones, meninges, and internal organs. And what I'll do later is I'll show you actually like molecule, which is considered to be the melanin molecule, and you can start to see how by it being an attractant molecule that is taking, receiving, giving a lot of input in the biological system, you can see how then contagions could be attracted to it. Valley fever is endemic to the desert valleys of California and the dry, dusty areas of southwestern United States, especially in Arizona, New Mexico, Texas, and part of Mexico. It is most likely that man and animals are infected by the inhalation of spore-bearing dust. And these are real significant things, because if you just even at home on a sunny day, you're sitting in your room, kitchen, anywhere in your house, and you see light coming through the house and the dust particles that are flowing, I mean, that dust is just there everywhere. Imagine spores that can be just released somehow and get into you. Well, that's what we're talking about in terms of these areas where it's primarily existing in these dusty places. There may be a reservoir of infection in small wild rodents, and these animals could pass the spores in their feces to contaminate the soil, after which the spores would be spread by the wind. The rather frequent dust storms of the Southwest might carry these organisms long distances. Do you remember a few years ago in the Midwest they had some rodents that had some disease? I don't know the exact name of it. I don't think this was it. But
Yeah, kill the Indian.
But what was, you remember the name of that? It's like these rodent feces.
Wipe out the Indian.
Yeah.
And then they tried to come up with a vaccine
For
the babies, but they infected the vaccine with AIDS.
Man. So that's the thing about, now we have to get to inoculations and start to question that thing. But when things happen and they blow it out of proportion, it might have been planned anyway, just to make people, alert of it. But they know exactly what they're doing, because you hype people, get them all alert and get them worried. Hey, now you set them up for what? Vaccinations,
I'm a diabolical.
Right. So you had something, you said you knew the same thing? And that thing about, so that's valley fever. So I'm just mentioning to you that there's documented evidence as to how it can have an impact. I told you about the Aspergillus fumigatus, and that can have a biological impact. Well, these are negative things. This, I think I have one more piece in this one. Yeah. This is just how the CIA may be thinking. In the late 1960s, the descendants of Sidney Lovell's Hellraisers had developed a gamut of chemical and biological devices suitable for every purpose, from disguised assassination to minor harassment. Some were described by former CIA agent Philip Agee in 1975. Horrible smelling liquids in small glass vials can be hurled into meeting halls. Just walk by, throw it in there, and keep on walking. Covert stuff. A fine clear powder can be sprinkled in a meeting place, becoming invisible after settling, but having the effect of tear gas when stirred up by the latter movement of the people. An incendiary powder can be molded around prepared tablets. When ignited, the combination produces ample quantities of smoke that attacks the eyes and the respiratory system more strongly than ordinary tear gas. A tasteless substance can be introduced to food that causes exaggerated body odor. And a few small drops of clear liquid stimulate the target to relax. And a few small drops of a clear liquid stimulate the target, that is the person, to relaxed, uninhibited talk. Meaning that you could probably make a person do things that they wouldn't necessarily do. And generally, that's what you think about alcohol. It's a depressant, but it causes inhibition, where you start saying things you know you shouldn't be saying under that alcohol. Invisible itching powder can be placed on steering wheels or toilet seats, and a slight smear of invisible ointment causes a serious burn to skin on contact. Chemically processed tobacco can be added to cigarettes and cigars to produce respiratory ailments. And talking to students, because when you ever talk about psychopharmacology, drugs, and all that stuff in class, they always got to talk about marijuana, because I don't know if they're doing it or not. But they talk about all these things now with chemicals that have been put in marijuana, and to either make it stronger or give it different effect. But you start talking about putting things in cigarettes, cigars, tobacco items, which then has a long-term impact. And that's why with that last piece, neurotoxins, over a long period of time, you start talking about having detrimental effects on the body's, adaptability. This thing about creating hype and getting people to think about, we got to get immunes, inoculated, vaccinated. This was an article in Time magazine about using technology for microchips, where they can put the little guy in the middle has a little teeny chip in his hand that can be inserted in the body, and that chip can be used to monitor health, from sugar levels if you have diabetes to maybe if you have some other hormone in your body that's imbalanced. This chip could be set up to test how that information is being distributed in your body. Also, maybe your social security number could be put on there and they could track you where you go. And I found this pretty devastating because when we talk about inoculations, the whole thing about smallpox vaccines. In the middle of Africa, the whole story, as many of you which should be educated to know already about, that was the impetus at the beginning as to how they got into the middle of Africa shooting people up, giving them smallpox vaccines. So people were saying that inside that vaccine may have been this virus that contaminated the individuals, and that's how it spread like that. I found this devastating, really, to even think about this in inoculations. I was just feeling my arm the other day, and 2 little bumps right here. Could be just from the, needle that goes in as a child. I mean, I haven't been inoculated since I've been an adult. The point is, as a child, 2 little bumps. I don't know if you even check out your arm if you got little bumps. It might just be the tissue from the injection, but it's quite obvious that things could be going on daily without us even knowing it, where they could put things in your body and you don't even know. So you wonder how somebody could bomb that, Oklahoma? Was it Timothy McVeigh bombed that place? And they found the fool a couple of days later? Was they tracking them? So the fact that they can't find certain individuals, but they can find some, you start to wonder, how does that happen? So we have to really be concerned about these inoculations and immunizations, even with our children that are growing up. And this whole thing about immunizations, we don't have an immune system, because we're not immune from nothing. We have a defense mechanism system. So we can probably give our bodies what it needs through our diet and nutrition to fight against things in defense. So these inoculations can be kind of dangerous. The polio virus has been something very significant for inoculations, meaning that it has helped to deter it. The polio virus enters the body just like AIDS does. So there's some connections between, again, all these inoculations, what they did in terms of studying that syphilis. Just trying to make the connections here. The 40 years of studying syphilis, what we know about as AIDS came along, and creating mass hysteria sometimes to make people want to get inoculated. And with African people being, I guess, the more gullible than others, we start to get caught up in some things that aren't necessarily to the best of our interest. This riot control, skin irritants, just a small piece on that. Riot control agents are basically peripheral sensory irritants. I mean, if you want to get some people, you get their eyes and skin, you make them real irritated, so what, they ain't fighting back and against you. So if they're studying the skin here, they're probably studying melanin and pigmentation, because that's something that's very attractant to molecules. Peripheral sensory irritants are substances that pharmacologically interact with sensory nerve receptors in skin. Some chemical warfare agents include agents that are blister agents, that is, actually makes blisters form on your body. Choking agents, lung irritants, blood agents, incapacitating agents. And if they know that they're going for certain populations sometimes, perhaps they use certain kinds of chemicals specifically targeted to that kind of people. So if we know that skin melanin has been studied for several years and still is, they have to have much evidence about how you can make those individuals' systems shut down quicker than other people's. If we know through these other diseases like valley fever and what is created from Aspergillus fumigatus, darn well that there's some ethnic variations now in the biology of people. This piece on alcohol and malt liquor. Has anybody ever questioned why we have malt liquor as a substance for us to consume?
Yeah. You notice that they advertise that all the time on B103 and black stations, but you never hear it on white stations like GST or WSB. You never hear anything about malt liquor.
And the word is never, huh?
You never.
Never.
But you hear it every 5 minutes on B103.
Mm-hmm.
I mean, that's been a question in my mind from day one.
Malt liquor, okay.
I remember back in high school, in 9th and 10th grade, on a country club and a Coke 45 came out. Some of my partners, we really experienced with it, and I find out today that they still projecting it right in the black neighborhood. This was in the early 60s. Those particular two malt liquors I know in Jordan that we was experimenting with.
Right.
As to where the regular premium, we wanted to get on up.
So the thing is, you hear on the commercials, just normal TV, basketball games, light beer commercials.
Yeah.
And that's usually marketed to white people. But they got the hard stuff, malt liquor, marketed to us. There were some problems with a chemical about maybe 8 years ago called Cisco, CISCO, and I guess it's probably still in the stores. It's like a wine drink that has 20% volume alcohol in it. It was creating these rambunctious feelings in these children where they were just doing some wild things and not killing one another, but doing some very bizarre acts. So a partner of mine stated, when I was working at Walter Reed, we need to somehow start taking our research and information to the people. Because here I am studying animals in a laboratory context, but not having any relevance to the experience that's going on with us as a people. So it's like, how can we somehow use the malt liquor study or information about it to look at it, how it's affecting people biologically? Well, as I just started reading some information about what's in these substances, it really doesn't tell you what's in these substances. But if you look at Wild Eyes Rose, Mad Dog 2020, those cheap wines that are in our communities, and you start looking at some of those things that are also in those wine drinks, it says contains sulfites, S-U-L-F-I-T-E-S, sulfites. Sulfites are used as preservatives. Even dried fruits, I think, have sulfites for preservatives. But sulfites aren't supposed to really be in your body. There's something called sulfite oxidase deficiency that exists in people. That's a problem. Sulfites, okay, something that shouldn't be in your body. Oxidase. Anytime you see a word that has ASE on the end of it, it's referring to an enzyme. So if you say sulfite oxidase, it's what? An enzyme that breaks down sulfites. So if you even got the wine stuff in your body and you're drinking it, okay, you at least have something in your body that can break it down. If it ain't there, you're going to get really drunk or really sick. So if you have a deficiency of that, you're going to have a problem consuming that type of substance. Now let's look at people who have a deficiency of that without even understanding or talking about alcohol as a consumption. In chapter 10 of Dark Matters, Dark Secrets, I wrote, physiologically, there is a severe problem with consuming the different types of alcohol that are marketed to low-income African Americans. For example, the cheap wines we just mentioned contains sulfites. Long-term consumption can potentially lead to neuropathological damage. What does neuro mean? N-E-U-R-O, neuro, brain. Pathological disease, tissue starts to die. For instance, McDonald and Johnson in 1990 have reported on the role of excitatory amino acids in developmental neurological disorders. Excitatory amino acids are something that the body does need to stimulate activity, so something you do need. An example of excitatory amino acid would be something like glutamate. And you heard of glutamate from maybe, I guess, Chinese food. Some people say no MSG, monosodium glutamate. Well, it's giving some people headaches, so it's stimulating things. So some people can't have it in their food. So that's what we're talking about.